Hi everyone, in the last lecture we talked about the anatomy and histology of the urinary system and went over the structure of the nephron. In this lecture we're going to start how blood flows into the nephron, and from there what are the steps of urine formation. So again, to recap the structure of the nephron, over here you are looking at the afferent arteriole through which blood is going to come into the nephron. Here you have the glomerulus, and this is the visceral layer of the glomerulus, so you're going to have podocytes and mesangial cells right there, and blood is going to exit the nephron via the afferent arteriole. Remember that the afferent arteriole has a wider diameter than the efferent arteriole. The efferent arteriole continues on to become the web of peritubular capillaries around the proximal convoluted tubule and the distal convoluted tubule. The blood capillaries also form a network around the loop of Henle, and this network of blood capillaries around the nephron loop is called the vasa recta. So the process of urine formation has three separate components. First, blood comes in to the glomerulus. Big substances of blood, such as blood cells, both white and red blood cells, plasma proteins, have to get caught in that sieve from the podocytes and go back into the bloodstream via the efferent arteriole. Now there is going to be fluid that gets squeezed out. So stuff that doesn't get caught in the sieve gets squeezed out here in your capsular space, which is where I'm drawing right now. So this fluid that just exited from the blood capillary and got squeezed out into the capsular space is no longer blood, nor is it urine yet. So this fluid, we are going to call it the filtrate. So that component of urine formation is called glomerular filtration. And we're going to go into each of these steps in detail. The next two components are happening concurrently. Let's talk about tubular reabsorption. So tubular reabsorption is when filtrate, which is right here, there's things out of the filtrate. So anything like nutrients that your body requires or needs to conserve, water, that kind of stuff goes from the nephron proximal convoluted tubule back into the bloodstream. It either diffuses across, it's facilitated diffusion, but when nutrients or solutes go from the proximal or the distal convoluted tubule into the bloodstream, it is called tubular reabsorption. Now there are wastes that don't get caught in the glomerulus. So the filtrate that is flowing through the proximal and distal convoluted tubules still has waste products, and that could include excess solutes. So that component of urine formation is called tubular secretion. So tubular secretion is when solutes go from the blood capillary and into either the proximal or the distal tubule. So from a nephron tube to blood tube is called tubular reabsorption. From blood tube to nephron tube is called tubular secretion. And this can happen at various stages in the nephron. So it's not just proximal and distal convoluted tubules. You can have secretion and reabsorption happening in the nephron loop and in the collecting duct as well. So let's look closely at the glomerulus, the visceral layer of the glomerulus here. What you're seeing here is a very large podocyte. So podocytes are amoeba-like cells. You see the big nucleus here of the podocyte, and you see little amoeba-like extensions here that are called pedicels. So you see the zipper-like formation. They're juxtaposed, and there's a zipper-like formation of pedicels. And the gap between these pedicels that you're seeing here, those are your fenestration slits. So essentially, as blood is flowing through this blood capillary right here, stuff or blood and blood products are going to go through the blood capillary. There are some substances that are going to get caught here in this sieve between the fenestration slit. And in the next step, some stuff is going to get squeezed out and does not get caught in the sieve. So we're going to see that right here. So you're looking at a very zoomed in version here. This is basically, this line right here is the blood capillary and the pores in the blood capillary. This is the pedicels, and you're seeing the fenestration slits in the green arrows right here. So passing through your bloodstream are your large white blood cells, red blood cells, platelets, as well as large proteins like albumins and globulins. But that can't go into the capsular space. So these pores are actually too small for any of this large material to pass through. So what does get squeezed out, what is called as the filtrate, it includes a lot of stuff that is either going to get reabsorbed back into your bloodstream later on, or it has wastes that are going to get secreted later on. So some of the things that are in filtrate is large amounts of water. About 99% of that water is going to get reabsorbed. It has glucose, which is a blood sugar. It has small amino acids, proteins, ions, urea, some hormones, and very, very small amounts of protein. So this includes stuff that you need, as well as stuff that eventually is going to get excreted out. So in a 24-hour period, we produce about 180 liters of filtrate. Now, if all of the filtrate were to become urine, we would be peeing out about 180 liters of urine a day, which would be ridiculous, right? So 99% of that filtrate ends up being reabsorbed into your bloodstream by the nephron, and we're going to look at that here in a little bit. Now, some material that's trapped, so there are some like bacteria or any sort of like dead cells or anything that ends up getting trapped in the pedicels or the fenestration slits, is taken care of by the macrophages that are present here, called the mesangial cells. So the mesangial cells are macrophages that go ahead and phagocytosize stuff that gets caught. Now, materials that are not basically squeezed out and become filtrate, they exit the renal corpuscle or the glomerulus through the efferent arteriole. And please note that the efferent arteriole has a smaller diameter coming as compared to the afferent arteriole. Now, one important component to basically look at normal kidney function is to determine net filtration pressure. So the word net here is key. So I'm going to start us off right here at the afferent arteriole. It has this larger diameter and blood is coming in right here. So the first concept here is glomerular hydrostatic pressure. It's denoted as an HPG or a GHP. Same thing. So this is the pressure that the fluid is coming in through, and it is going this way or this way through the glomerulus. However, there are two opposing pressures here. The first of two opposing pressures is the blood colloid osmotic pressure. So this is the pressure that is a counter pressure because all of the colloids, the larger proteins in blood, are exiting through the efferent arteriole, and this pressure is exerted this way, opposite to your glomerular hydrostatic pressure. Now, this space right here is your capsular space, right? So as fluid is squeezed out from your fenestration slits, this sac or this capsular space is going to start getting all filled up with filtrate. And as a reason from fluid being filled up in it, there's a pressure that builds up. So that pressure that builds up inside your capsular space is called capsular hydrostatic pressure. So typically the mean blood pressure of blood going through the afferent arteriole is about 60 millimeters of mercury. Now the two opposing pressures here, the blood colloidal osmotic pressure ends up being 32 millimeters of mercury, and the capsular hydrostatic pressure 80 millimeters of mercury. So if you were to imagine like your gross income minus taxes would be your net income, right? We're not doing income here, but this is gross pressure. The glomerular hydrostatic pressure is your gross or incoming total pressure. You're losing the federal income tax, and then you're going to lose the state income tax. So you're taking out two pressures because they're counter pressures to give you a net filtration pressure. So let's look at how that is calculated right here. So what's happening here, so the net filtration pressure is off the fluid that is going to enter your proximal convoluted tubule. So essentially, GHP or HPG, both are acceptable ways to refer to it, is basically the blood pressure in the glomerulus itself. Now this is higher than blood pressure systemically. So when I say systemic blood pressure, we're talking about all the other arteries in your body. Now, we require a higher pressure here because you want to squeeze stuff out. So it's like a coffee filter. You press on the coffee filter and that's when liquid comes out. That's the higher pressure here. And that is achieved by having the incoming arteriole, which is the afferent arteriole, be a larger diameter. And then the outgoing arteriole, which is the efferent arteriole, have a smaller diameter. So if there's a hose, which is the afferent arteriole, and you want to pressurize stuff going out, you basically just pinch the hose and stuff starts coming out with higher pressure. It's that sort of a logic here. So larger diameter of afferent arteriole, it gets pinched as it's leaving, so that's how you get higher pressure. Now the two pressures opposing that I mentioned as federal income tax and state income tax, so these are things that we are going to subtract out of the incoming pressure, is your blood colloid osmotic pressure. So this is osmotic pressure by particles, larger particles like plasma proteins that are in blood. And this opposes filtration, right? So it's preventing things from getting filtered. And these large particles basically draw fluid back into the glomerulus. So they're not, this pressure here, the blood colloid osmotic pressure, is not contributing towards making the filtrate. It is an opposing pressure or counter pressure, if you will. The capsular hydrostatic pressure, the CHP or the HPC, is basically the pressure that builds up in the capsule or the capsular space as filtrate starts to build up in it. So here we go. So net filtration pressure is calculated by taking the glomerular hydrostatic pressure, which was 60 milligrams of mercury, and then subtracting the two opposing pressures here, 1 and 2. So the two opposing pressures here are blood colloidal osmotic pressure and your capsular hydrostatic pressure, which are 32 millimeters of mercury and 18 millimeters of mercury. So if you add the stuff in the parentheses, it is 50 millimeters of mercury, which is what your tax would be. This is your gross income, right? So 60 minus 50 equals 10 millimeters of mercury. So 10 millimeters of mercury is typically what the net filtration pressure is. So this is the pressure at which the filtrate is formed and the filtrate going into the proximal convoluted tubule. Now the net filtration pressure is important because that is what feeds into another very important variable in terms of the urinary system. That variable that we're going to discuss is glomerular filtration rate. So rate is basically an event, or how many times an event happens against a unit of time. So against a minute, an hour, a day, and so forth. So basically the definition of GFR is the rate at which the volume of filtrate is formed per minute. So how much filtrate is formed per minute is basically your GFR. And so here's what could happen. When net filtration pressure goes up, when net filtration pressure increases, that increases GFR. And we maintain GFR as homeostasis. So there's a range of normal GFR. GFR is supposed to remain constant no matter what your hydration status is, no matter what kind of stress you're in, because GFR can tell you if your kidneys are functioning normally or if you're starting to show signs of kidney disease or kidney failure. So increased net filtration pressure will make more urine, right? So it increases your glomerular filtration rate, and it increases the amount of solutes and water that are flowing through the tubular fluid or filtrate. So what ends up happening is that it increases substances in the urine. So you're losing stuff that you would normally reabsorb when GFR goes up. You're losing important solutes like sodium, potassium, calcium, bicarbonate, glucose, things like that. You're losing that when your GFR goes up, and as a result you could have ion imbalances, hypoglycemia, and so forth. Increased GFR also decreases the filtrate reabsorption rate. So the lesser is reabsorbed, the more it is peed out and ends up in urine. So for that reason, no matter what your hydration status is, if you go an entire day without drinking water, your body is still going to need to process and remove wastes, and your nephrons are still going to have to make urine to get rid of those wastes, even though you haven't hydrated that day. If you're well hydrated that day, you're going to pee out the excess water, but your GFR should remain constant no matter what your hydration level is. So many things can throw GFR off. Dehydration versus overhydration, different types of stress, right? Stress can throw GFR off as well, but your body will try to regulate. It will try to fix. If your GFR changes mildly, your kidneys will try to take care of that problem and fix that. If that fails, your brain comes into the picture and it becomes like a body-wide physiologic process that is also going to fix your changed GFR. Your GFR has to remain constant. There is a homeostasis of GFR. Now, there are two factors that go into determining the GFR, right? Width of or diameter of the afferent arteriole. So the larger the diameter of the afferent arteriole, the more blood that passes through it. The more blood that passes through the afferent arteriole, the more filtrate is formed. The opposite is true as well. Less the diameter of the afferent arteriole, less blood passes through it. Less blood passing through it means less filtrate is formed. The other factor that goes into this is the surface area of the filtration membrane, which is your podocytes. So there are times when those podocytes and mesangial cells can sort of crunch up and lose surface area. So they prevent more filtrate from forming. Now, say something threw your GFR off. First, let's talk about hydration status, right? You become dehydrated. You're dehydrated, but you still need to make urine because wastes are still produced and you have to excrete those wastes. So first, your kidney, your GFR went down, your kidney is going to try to fix it in-house. Those are the intrinsic controls. If that fails, a body-wide, like several organ-wide physiological process happens to kind of bring your GFR back to normal. And that is called the extrinsic controls. Now, here's what happens. So if you're dehydrated, your blood volume goes down. And when blood volume goes down, your blood pressure goes down. So on the x-axis here, you are looking at mean arterial blood pressure. So the blood pressure, the mean blood pressure in your arteries in millimeters of mercury. So anything from 80 to 180 is normal. So say you're really, really dehydrated. You haven't hydrated in a couple of days. You're lost in the desert. Something happens and you have not replenished on water. So as a result, your blood volume decreased, and because your blood volume decreased, your blood pressure decreased. So when blood pressure decreases, so does GFR. So GFR is right here on the y-axis, and normal GFR is about 90 liters per day. That's filtered. That's not the amount that you are peeing out. That's the amount of filtrate produced per day. So if you're dehydrated, GFR drops down. So if something happens and you're overhydrated, or something happens and your blood pressure, or you're stressed, let's say you're stressed, your blood pressure goes up, your blood volume increases. Your blood volume increasing is going to increase your blood pressure. So when blood pressure increases, so does GFR. And here are the consequences. So when GFR decreases, you're not making enough urine. When you're not making enough urine, the wastes are still circulating in your blood. That's the consequence. The ultimate consequence of insufficient urine production is that there are wastes that are accumulating in your circulating blood. Now, when GFR increases, you're having excess urination, and that's when you're losing ions, you're losing water, and you could feel things like lightheadedness, dizziness, and so forth. So let's look at how the kidney tries to fix GFR, or it tries to maintain GFR in-house. So I'm going to start with image B here. because this is what happens when systemic blood pressure is normal, right? So when systemic blood pressure is within that 80 to 180 range, blood comes through the afferent arteriole. It goes into the glomerulus, which is where the podocytes catch all of the blood cells, the larger proteins from the plasma, and that goes back out with high pressure out the efferent arteriole. Now it gets squeezed out into the capsular space. That's your net filtration pressure, and off it goes into your proximal convoluted tubule. Now let's see what happens if you had water loss. So you're dehydrated, you have decreased blood volume, because whatever water you're drinking, it's actually going to go into blood. So decrease of blood volume equals decrease of systemic blood pressure. Now, when your blood pressure decreases, what's happening is that less blood is going into the glomerulus to begin with. That's what's happening. So less blood going into the glomerulus means that less filtrate is produced. And that's what is throwing off your GFR. It is decreasing your GFR because low incoming blood equals low amount of filtrate produced. So the kidney tries to fix it by vasodilation of the afferent arteriole. So your systemic blood pressure decreased, right? You got low blood pressure. Here in the afferent arteriole, you're going to see a dilation. So when it dilates, more blood is going into the glomerulus. So here what's happened is your GFR has gone below normal range, and you want to bring it back up to the normal range. So now that the afferent arteriole has widened, more blood is gushing into your glomerulus. More blood going in equals more filtrate being formed. And this filtrate is going to go through the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, through the collecting duct and become urine. So what you're achieving here is you're maintaining GFR, so all the wastes that are circulating in your blood have a chance to become urine and get excreted out of the system. So this is how it fixes GFR if dehydration or low blood pressure suddenly manages to decrease your GFR. Now, here's the opposite scenario. You're stressed and your systemic blood pressure went up. It's above that 180 range that we saw in the previous slide. So normally, if blood pressure increases, there is going to be more blood that is obviously going to go into the glomerulus. The fix here is for less blood to go into the glomerulus, right? That's the fix. So if your GFR is too high, we're going to do something to decrease that and bring it back down to the normal range. So here's what happens. So in this case, if increased blood pressure ends up shifting your GFR temporarily, makes it higher, the afferent arteriole vasoconstricts. So it constricts a little bit, so less blood going in equals less filtrate being formed. Less filtrate being formed equals less urine being formed. The additional thing that happens here if GFR goes high is that your mesangial cells contract, they scrunch up and decrease the surface area too, right here in the glomerulus. So middle is homeostasis. If your blood pressure goes down, your blood volume goes down, your GFR goes down. Your kidney fixes it by vasodilating the afferent arteriole. More blood go into the afferent arteriole, more filtrate gets produced. More filtrate gets produced, you're able to excrete your waste in urine. When you're stressed and your systemic blood pressure is high, what the kidney is doing is vasoconstricting your afferent arteriole. So less blood going in equals less filtrate being formed, less filtrate being formed equals less urine, and thereby it decreases glomerular filtration rate. So the upper level, so that was the first level of maintaining GFR in the normal range, right? So if that fails, then neural and hormonal control, which are extrinsic controls, they don't happen or don't start in the kidney, start to happen. So the first way, so your kidney, this involves changing GFR. So what autoregulation did was it maintained GFR in the normal range. So what the extrinsic controls are doing, that they're actually changing your glomerular filtration rate. So if your GFR went high, right? So your blood pressure got too high, which is why there's more blood going into your nephron. More blood going into your nephron equals more filtrate, and therefore you have increased GFR. Now to fix that, you have to decrease GFR. And that happens by sympathetic stimulation. Remember, sympathetic is fight or flight. So essentially, you are conserving water when you are under stress. So you're exercising, you're stressed, you have an emergency going on. That will result in decreased GFR for a lot of different reasons. First, vasoconstriction happens of the afferent and or the efferent arterioles. And then the granular cells of that juxtaglomerular apparatus. So in the previous lecture, we talked about the juxtaglomerular apparatus, which was wedged. It had the JG cells and the cells of the macula densa that were wedged between the afferent arteriole and your distal convoluted tubule, right? So this actually ends up releasing something called renin. And renin is a whole, what follows is a whole system known as the RAA, RA system. So you want to conserve water here. You want to decrease GFR and therefore conserve water. So let's see how this happens. Now, there was a stressor or an emergency. Your blood pressure went up. Because your blood pressure went up, your GFR ended up going above your normal range. Our job here is to decrease GFR and bring it back into the normal range. So in this particular flowchart, we're talking about the insult or the exterior environmental change that ended up increasing your GFR above normal. And as a fix, your job is to decrease GFR by sympathetic, which is fight or flight stimulation. So there's sympathetic stimulation of the kidneys, and here is what ends up happening. First, the afferent arteriole constricts. Less blood going in equals less filtrate forming. Less filtrate forming decreases GFR. Here, the mesangial cells also contract, thereby decreasing surface area. So lesser surface area equals lesser filtrate produced as well. The other pathway is where cells of the juxtaglomerular apparatus release a hormone called renin. And renin is going to then produce, via a pathway, something called angiotensin. So let's look at what that is. So this entire system is called the RAA system. So let's start with the initial stimulus. The initial stimulus could be stress, it could be dehydration, it could be blood loss. So essentially blood volume went down, therefore blood pressure went down. Now here there was decrease in blood volume, there was decrease in blood pressure. The juxtaglomerular cells of the kidney secrete something called renin. Now when there's renin in the system, that is stimulating the liver to secrete something called angiotensinogen. And you know from the digestive system chapter that anything that ends with a gen is probably a precursor. So renin and angiotensinogen together convert angiotensinogen to something called angiotensin 1. So let's recap up to here. Something happened, you lost blood, or you're dehydrated, blood volume went down, blood pressure went down. Therefore, your GFR went below normal. It is the opposite scenario that we saw in your previous slide. In your previous slide, we saw that GFR went up and we were trying to decrease GFR. In this scenario, it is the opposite that's happened. Your blood volume went down, your blood pressure went down, as did your GFR. So your job here is to increase GFR until it gets into your normal range. Now, the decrease in blood pressure is actually sensed by the juxtaglomerular cells in the kidney itself. Then they're going to secrete renin, which stimulates the liver to secrete angiotensinogen. Now, angiotensinogen, in the presence of renin, converts to something called angiotensin 1. Angiotensin 1 is still not the final product. It's going to need one more component that is going to come from the lungs. And that component, that enzyme that it needs, is called angiotensin converting enzyme, also known as ACE. So ACE is converting angiotensin 1 to its active form, which is angiotensin 2. Now, angiotensin 2 is floating around in your blood, and this is what it is doing. It goes and causes vasoconstriction of your arterioles. So it's like cinching the hose, and when the hose is cinched, there's blood pressure. The fluid is going through it with higher pressure, right? So therefore, it is temporarily increasing your blood pressure. Concurrently, here's what's also happening. That adrenal gland that sits atop each kidney is secreting a hormone called aldosterone. So aldosterone is doing this. It goes to the kidneys and it is stimulating the proximal and the distal convoluted tubules to increase reabsorption of both sodium and water. So essentially, it's preventing water loss. So if you already have low blood pressure, you don't want to pee more, because not only are you going to lose water, you're going to lose blood volume even more, and that will only further decrease GFR. In this case, we want our GFR to go back up into the normal. So here it is going to tell the nephron to send sodium and water back into blood. So that is how blood volume is increased until blood pressure goes back to normal. And when blood pressure goes back to normal, your GFR goes back to normal. So the first R here stands for renin, which is secreted by the kidney itself. Then you have the angiotensinogen, the final form, the angiotensin II, and the aldosterone. So that is the Ra system. And the Ra system is kicked into, or it's stimulated when your GFR has gone down because decreased blood volume and decreased blood pressure. Now, if your renal autoregulation fails, like we saw a couple of slides ago, which is where GFR went up because blood pressure went up and you ended up contracting your afferent arteriole. So here is how that happens. Now, blood pressure increased, right, because of stress, or blood volume increased because you drank a lot of water. The atrial wall stretches, right? So there's more blood volume, so all blood has to go through the pump, the heart. So when there's more blood volume, the walls of the atrium, which is the top two chambers of your heart, that stretch. And when that stretch is detected by the heart, it's going to secrete something called ANP, atrial natriuretic peptide. So here is what it does. The atrial natriuretic peptide is going to go and cause vasodilation of your afferent arteriole. More blood going in equals increased GFR. So I'm going to stop here and we're going to pick it up again and talk about how urine is formed as things are going through the proximal and distal convoluted tubules.